Hello everyone. Today I am going to make you learn about DNA replication. DNA replication is very important process for all the living system. This topic is from the chapter Molecular Basis of Inheritance, class 12th textbook of biology. So, in this session we will learn about DNA replication and its importance, different models of replication, Meselson-Stahl experiment, types of DNA replication, various enzymes or proteins involved in DNA replication and mechanism of DNA replication. So, let us start the session. What is DNA replication? As we know, replication is a process of making any copy or making a replica. Replica means exact copy. So, DNA replication is a process by which a double stranded DNA molecule is copied to produce two identical DNA molecules. One of the DNA molecule, if suppose here it is a DNA molecule and if it is copied to form two DNA molecule, this process of making the copy of DNA is called DNA replication. Now, here an important question is arise, why DNA replication is so essential, why it is required by all the cells, all the living system. So, DNA replication is required for cell division, for reproduction and for synthesis of RNA and protein. As we know, every cell divide to form two daughter cell in case of mitosis, and in case of meiosis the cell divide to form four daughter cells. But have you ever think how a cell divide and the new daughter cells have the same number of chromosome as that of the parental cell in case of mitosis? Suppose a cell is there and as we know cell is having 46 number of chromosome. In a general way, if we think about it, if a cell divide and it is forming two daughter cells, so division means this chromosome should get halved if it is, if we are thinking about mitosis, but it is not like so. This is the incorrect concept. The actual is that in case of mitosis, the parental cell will divide to form two daughter cells with same number of chromosome. Now, how it can be possible? The daughter cell is also having 46 chromosome and parental cell is having 46 chromosome, and this is because of the replication which occurs during the S phase of cell cycle. And synthesis of RNA and protein, in the later session we will learn about transcription and translation, where DNA will be copied into the RNA and then messenger RNA will form the protein. Now, three models have been proposed for replication. The first was conservative model, second one was semi-conservative model and third one was dispersive model. Now, what these models are? So, in case of conservative model, if it is a parental DNA, this parental DNA will be copied as such without any change to make two daughter DNA. You can see after replication one DNA, daughter DNA is exactly same as that of the parental DNA and another daughter DNA is copied of parental DNA, and this is the conservative model of replication. Second model was semi-conservative model, in which the two parental DNA strands separate from each other, separated strands then serve as a template for the synthesis of new DNA strand. The result is two DNA double helix, both of which consist of one parental DNA and one new strand. Let us understand in this way. If it is a parental DNA, in case of semi-conservative model, both the strands of the parental DNA will get separated out and these separated strands of the parental DNA will act as a template, and this template will be copied. So, each of these daughter DNA will have one old strand and one newly synthesized strand of the DNA. And the third model was dispersive model. In this dispersive model, it was expressed that the parental DNA will get fragmentized and each fragments of the DNA will act as a template to make the new strand of the DNA, like this. So, these are the three different models proposed by the scientist: conservative, semi-conservative and dispersive. In case of conservative, the parental DNA will copied as such to make two daughter DNA. In case of semi-conservative model of replication, the parental DNA will get separated out and the separated single stranded DNA will act as a template, and this single strand DNA will be copied in both the daughter DNA and they will form two daughter DNA. And in case of dispersive model, the parental DNA will get fragmentized and each fragments will act like a template, they will be copied to make two daughter DNA. So, there was a great confusion among the scientists ki out of these three by which model actually the DNA is being replicated. So, here are the some picture related to this model of replication. This is the semi-conservative model. You can see this parental DNA, both the strands get separated out and new strands are being synthesized. In conservative model, this parental DNA will remain as such and it will be copied to make two daughter DNA. And in dispersive model, the parental DNA will get fragmentized and each fragments will be copied to make two daughter DNA. Now, to solve the mystery ki how the DNA get replicated by conservative or semi-conservative, the two scientists Matthew and Meselson and Franklin-Stahl in 1958 they proved experimentally that DNA replication is semi-conservative mode of replication. What they did is they cultured bacteria in N15 media. As we know ki normally all the DNA having the N14 in their nitrogenous bases. They culture the E. coli cell in N15 media, and N15 media is radioisotope of nitrogen having slighter greater density than that of the N14. After growing the E. coli cell in N15 media for a longer period, they transfer the cell on the fresh media having the nitrogen N14, and then after each cycle they isolated the DNA from bacteria and then they separated the DNA on cesium chloride density gradient centrifugation. Now, they found the result just same as that of the semi-conservative model. We can understand this experiment like this. Meselson and Stahl, they grew E. coli cell these are E. coli cells. E. coli cells are bacteria and they took N15 in media which is a radioisotope of nitrogen. After culturing the cells for many generations, they transfer the cells in another fresh media having nitrogen N14. Now, from this media they isolated the bacterial cell after each generation to check whether the DNA get replicated according to conservative or semi-conservative mode. And again in second cycle of replication, they took cells, isolated the DNA and separated the DNA on cesium chloride density gradient centrifugation. So, this here we have parental E. coli cell in which nitrogen is N15, and in this newly synthesized E. coli cell new DNA will be having N14 and here also N14. When they took the DNA from the parental E. coli cell and separated the DNA on cesium chloride density gradient centrifugation, they found that there was a single band which correspond to the density of N15. As we know कि parental E. coli cell were having N15 in their DNA. After first cycle of replication, when they isolated the DNA from the E. coli cells and separated the DNA on density gradient centrifugation, they found that there was again one band which correspond to the density of intermediate of N15 and N14. And in second cycle, again they isolated the DNA from bacteria and separate the DNA on cesium chloride density gradient centrifugation, and they found two bands of DNA which corresponds to, the lower one corresponds to intermediate of N14 and N15 and the upper one with N14. Now, this result was according to conservative model. Let us see how it can be. If suppose DNA is being replicated in semi-conservative model, then what would be the result? This is a parental DNA. Parental DNA is having N15 nitrogen in both the strands. After replication, both the strand will get separated and new strand will be synthesized. The new DNA will be having N14 and the older is already N15. This result is after first cycle of replication. If we go for the second cycle of replication, all these 4 strands will get separated and they will be copied. So, in this one is the older one having N15 and all the newly synthesized will be having N14 nitrogen. So, if we go for the cesium chloride density gradient centrifugation in parental DNA, we will get just one band which is of N15 and it is in lower side. If we go for the first cycle of replication and if we take out the cesium chloride density gradient centrifugation, we will find just one band with intermediate of N14 and N15, and in second cycle we will get two bands. As we can see here, two DNA molecule have the intermediate of N14 and N15 and two DNA is having newly synthesized DNA with all N14. So, here the result will be one band of N14 and another band will be intermediate of N15 and N14, and this result was exactly same as that obtained by Meselson and Stahl. So, hence they proved that this DNA replication is semi-conservative mode of replication. We can see the result in conservative also. Suppose consider that DNA replication occurs in conservative mode, then what would be the result? If it is a parental DNA, it will be N15 N15, and after first cycle of replication the parental DNA will remain as such, that is N15 N15, and it will be copied to form another DNA with N14 N14. This is after first cycle of replication, and if we go for second cycle of replication, this process will be same. The parental DNA for second cycle, this will be the parental DNA, will remain as such, and two new daughter DNA will be synthesized. One DNA will have N15 N15 and all the newly synthesized will be having N14 N14. And if we go for cesium chloride density gradient centrifugation, we will get one band of parental DNA as N15 in a lower side, because N15 is slightly dense than that of the N14. After first cycle, if DNA is being isolated from the culture, and if we go for cesium chloride density gradient centrifugation, in conservative model there will be two bands, one will be of N15 and another will be of N14. After second cycle of replication, we will again get two bands, one will be of N15 and one band of N14. So, here the result was totally different from the result obtained by Meselson and Stahl. The result of Meselson and Stahl was correspond to the conservative model, hence they proved that DNA replicate in conservative model, in which both the strands of the DNA first get separated out. Now, this separated strand will act as a template, they will be copied and then two daughter DNA will be synthesized. Now, types of DNA replication. There are three main types of DNA replication in which the DNA is being replicated. The one is theta replication. In case of theta replication, If suppose DNA is circular in case of E. coli cell and here it is oocyte, oocyte means from where the replication will begin. From this oocyte, both the strands will get separated and from this separated strand the DNA will replicate. The structure seems to be like theta, so that is why it is known as theta replication. Next type of replication is rolling circular replication. In case of rolling circular replication, if it is a circular DNA, one cut will be made in one of the strand of the DNA and that strand will get separated from the main DNA and this gap of DNA will be copied like this and the new 2 DNA will be synthesized. And the last type is linear eukaryotic replication, in which this DNA will be replicated at multiple sites wherever the oocyte will be there. Suppose here we have oocyte, here we have oocyte. So, at both these sites the DNA will get separated like this and the DNA will get replicated at multiple sites. This type of replication mainly occur in eukaryotes. Now, what we need, what the cells need for the replication process? The cell need a template which will be copied to form 2 daughter DNA. The cells need raw materials, that is dNTPs. As we know, DNA is having 4 type of dNTPs: dCTP, dGTP, dTTP and dATP. These dNTPs will act as a substrate during the polymerization of DNA, and various enzymes which will help the cell to synthesize 2 daughter DNA. Now, what are the different enzymes or proteins which involve in replication? Here are some of the enzymes. The one is DNA helicase, which help in the separation of double stranded DNA. Primase enzyme, which is actually a RNA polymerase enzyme, which synthesize a primer, which we discuss later in this session. Next enzyme or protein is single stranded binding protein. It is mainly to stabilize the single stranded DNA so that it cannot reanneal after its separation. DNA polymerase, without which the DNA cannot be replicated. So, polymerization of DNA will occur only with the help of DNA polymerase. Topoisomerase, which is one of the type of DNA gyrase. Topoisomerase, when the DNA is being separated out, the other sides of the DNA will get supercoiled, and supercoils may inhibit the process of DNA replication. So, to reduce the supercoilings, the tension, the torque exerted by the supercoilings, this topoisomerase will involve, and DNA ligase, which finally creates a phosphodiester bond during the DNA replication. Now, this is an important part of DNA replication, direction of replication. Most of the children are getting confused in which direction the DNA will be replicated, whether it will be from 5 to 3 or 3 to 5. So, you must recall the concept which I made you learn in my earlier session. If suppose it is a nucleotide, here the first carbon, and at first carbon any base will be there, nitrogenous base, at second no H is there, that is why it is deoxyribose sugar. Here we have OH at 3 number of carbon, and phosphate at 5 number of carbon. This is a general diagram of DNTP nucleotide. So, you can see over here कि at third number of carbon only it is having OH, and because of this OH any nucleotide will get added over here only. So, if we talk about direction, here we have 5 prime end and here we have 3 prime end. So, DNA replication will always occur in 5 to 3 direction. You can see the new nucleotide only bind over the 3 end, because 3 end will have free OH group and phosphodiester bond will be created over here only. So, this question might be clear to you ki how, in which direction DNA will be replicated, whether it will be in 5 to 3 direction or 3 to 5 direction. Now, mechanism of replication, how the replication is being completed, what are the different steps? The one is initiation of replication, second one is elongation of DNA chain, that is polymerization of nucleotides, and then termination. If any process is getting started, it means it will be terminated somehow. Now, as a model we will learn about the process of DNA replication in bacteria. In case of eukaryotes, many of the things are very much similar as that of the bacterial DNA replication. Later we will learn about the differences in eukaryotic and prokaryotic DNA replication. So, initiation of replication. In case of bacterial DNA, only one oocyte is there. This is a major difference between eukaryotic and prokaryotic DNA replication. Eukaryotes have only one oocyte. Oocyte means from where the replication will begin, and in case of eukaryotes there are many oocytes, and from almost all oocytes the DNA can get replicated. Suppose it is a prokaryotic DNA, and here we have oocyte. Now, at this oocyte one of the initiator protein will bind and it will help to unwind short section of DNA, and then these short section of DNA will now unbinded by the helicase enzyme. So, first of all an initiator protein will bind over the oocyte and made the DNA unwind in small fragments. So, this unbinding of short stage of DNA is because of the initiator protein, and after this unbinding DNA helicase enzyme will continue the unbinding of DNA strand. Two DNA helicase will involve, and will unwind the DNA in both the direction. Now, this single stranded DNA will act as a template and then they will be copied by DNA polymerase to make two daughter DNA. But here comes an important problem. As we know कि DNA is having the nature to make double stranded DNA, to be in double stranded. So, these separated strands will not be stable for a longer period. To get them stabilized, and another protein called single stranded binding protein will bind over the template and will stabilize the single stranded DNA, so that it will remain in single stranded for the DNA polymerase to make it copied. Now, when the DNA is being unwinded, when the being is separated during the replication, these supercoilings are being formed in the DNA, and these supercoilings may inhibit the process of DNA replication. So, how these supercoilings can be removed? These supercoilings will be removed by the enzyme topoisomerase, one of the DNA guidase enzyme. This enzyme reduces torque or tension by making a double stranded break in one segment of the DNA helix, passing another segment of the helix through the break and then reselling the broken ends of the DNA. That means, this topoisomerase will cleave one of the strands of the DNA, so that this torque, this tension can be released and the DNA supercoilings can be removed. Now, here comes another important question. As all DNA polymerases require a nucleotide with a 3-end OH group to which a new nucleotide can be added. As I explained earlier, the DNA replication is always in 5 to 3 direction, and this particular nucleotide is having OH at 3 end. This 3 end is required by DNA polymerase to add the new DNA. Without this 3 end, the DNA cannot add the nucleotides. Now, how this 3 end OH group is being provided to the DNA? And this problem of DNA polymerase is solved by an enzyme called primase. Now, what is this primase? Primase is one of the type of RNA polymerase. The detail of this enzyme we will learn in the chapter transcription, but here I should tell you this RNA polymerase is able to synthesize RNA nucleotides without the help of any free 3' end, without the help of 3 OH group. Because of this reason, this primase enzyme will synthesize a short segment single stranded oligonucleotides and provides a 3 free end to the DNA polymerase, so that the DNA polymerase can bind nucleotides at 3 end. So, the primase enzyme will bind, if suppose it is a 5 end, 3 end, so here it will be 5 end and it will be 3 end, and here it will be 5 end or 3 end. So, these short segments of oligonucleotides synthesized by primase enzyme is called primer. Now, this primer will provide 3 free end in both the template to the DNA polymerase to bind the new nucleotides, and then the DNA replication will begin. Now let us discuss about the elongation process, how the DNA is being elongated. Consider the second half of DNA which is already separated. This separated strand is appear like a Y, and that is why it is called Y fork. And in both this separated strand, which is we call template, the primer will get attached, and suppose this is a primer 5 end and 3 end. Here also the primer will get attached. Now in this particular strand, now this primer is providing 3 free OH group or end for DNA polymerase to bind the new nucleotide. Now DNA polymerase will bind over there, it will read the nucleotide of the template and will add the new nucleotides which will be complementary to the template. So here the replication will always in 5 to 3 direction, and the replication is continuous. That is why this strand is called leading strand. But in case of this strand, here it is the primer and this primer is providing 3 end over here. Now this fragment of the separated strand will be copied by the DNA polymerase. Again another primer will bind wherever they will get the complementary 3 end, 5 end, and these gaps will be filled up by DNA polymerase 5 end and 3 end. Now like this whole the strands of the DNA will be replicated, but do you think after the formation of both the strands and both the daughter DNA, the daughter DNA or the newly synthesized DNA are exactly the same as that of the parental strand? Yes, it is not exactly same, because these primers are not the exact copy of DNA template. These are the primers having ribonucleotides, means they have uracil nucleotide instead of thymine nucleotide. So these primers are being removed by an enzyme, one of the type of enzyme DNA polymerase 1. In case of bacteria, in case of prokaryotic cell, there are 5 types of DNA polymerase. They are DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4 and DNA polymerase 5. Out of these 5 DNA polymerases, only 2 DNA polymerases involves in DNA replication, and they are DNA polymerase 1 and DNA polymerase 3. DNA polymerase 2, 4 and 5 involve in DNA repair. Whenever there will be some damage in the DNA, that will be repaired by all these 3 DNA polymerase. Now DNA polymerase 1 will remove the primer. Now what will happen if primers are being removed? Let us see if DNA polymerase removes the primer, what will be the scenario? Three end, 5 end, 3 end, 5 end, and here there is a gap, and it will be 5 and 3 end. Now what about these gaps? how it will be filled, and if it is kept unfilled, then it will be not a proper DNA replication. Now, these gaps will be filled up by DNA polymerase 3 taking the 3 free end of this newly synthesized DNA, and these newly synthesized DNA in fragments are called as Okazaki fragments, and this strand is called lagging strand. Now, these Okazaki fragments will give 3 free end to the DNA polymerase to fill the gap. Now, this gap will be filled up, but last nucleotide, here we have 3 end and here we have 5 end. These two nucleotides cannot be form phosphodiester bond because DNA polymerase cannot help to form phosphodiester bond. So, here an enzyme called DNA ligase, this DNA ligase will involve over here and it will form the phosphodiester bond in between this 3 and 5 end, so that the DNA strand will be completed, and accordingly the whole this DNA will be copied to make 2 daughter DNA strands. So, DNA polymerase 3, they have two important function. One, its 5 to 3 polymerase activity, which allow it to add new nucleotides in 5 to 3 direction. As I already told you, the DNA replication occurs in 5 to 3 direction, and this 5 to 3 polymerization of DNA on nucleotides is promoted by DNA polymerase. And another important activity of DNA polymerase is its 3 to 5 exonuclease activity. Suppose DNA polymerase adds any wrong nucleotide, here we have any wrong nucleotide. Now, these wrong nucleotides will be removed by its exonuclease activity and the correct nucleotide will be added. So, these two important feature of DNA polymerase make the DNA replication accurate and fast. So, this already we have discussed. The removal and replacement of primers appear to constitute the main function of DNA polymerase 1, that I already told you that DNA polymerase 1 removes all the primers and then those gaps will be filled by the DNA polymerase, and DNA polymerase 2, 4 and 5 function in DNA repair. Here is the complete description and function of DNA polymerases of prokaryotes. Now, let us summarize the function, the activity performed by DNA polymerase. DNA polymerase synthesize any sequence specified by the template strand. The DNA will add only those nucleotides that will be complementary to the template. The DNA polymerase synthesize in 5 to 3 direction by adding nucleotide to 3 OH group. The DNA polymerase add the nucleotide at the 3 prime group because at 3 prime end they have OH group and phosphodiester bond will be created over there only. DNA polymerase use DNTPs to synthesize new DNA. Without DNTPs, new DNA cannot be synthesized. DNA polymerase require primer. Without primer, DNA polymerase cannot initiate the replication, cannot participate in polymerization, and these primers are synthesized by DNA polymerase enzyme. DNA polymerase catalyze the formation of a phosphodiester bond by joining the 5 end phosphate group of the incoming nucleotide to the 3 OH group of the preceding nucleotide on the growing strand. It means that the old nucleotide is having free 3 OH group, where the new nucleotides will be added with its 5 phosphate group by forming the phosphodiester bond, and DNA polymerase produce newly synthesized strand that are complementary and antiparallel to the template strand. So, new strand which will be synthesized, which will be polymerized by DNA will be complementary and anti-parallel. If one strand is 5 to 3, the new strand will be 3 to 5. Now, important, which we generally skip from the syllabus, the termination of DNA replication. In case of prokaryotic DNA replication, there is a two ways of getting DNA replication terminated. One, when the two fork meets together. If both the forks meet together, the replication will be terminated. And another specified by termination sequences, where a TUS protein will be added, and this TUS protein will inhibit the movement of DNA helicase, and because of that the DNA replication will be terminated. Now, let us summarize all the concepts which we learned till. Helicase enzyme which unbinds the DNA, single stranded binding proteins to keep the nucleotide strands separated long enough to allow replication, the topoisomerase, one of the type of gyrase enzyme which removes the supercoalinks formed ahead of the replication form, the primase which synthesize primers and provide 3 OH group to the DNA polymerase so that the polymerization can be done. DNA polymerase to synthesize the leading and lagging nucleotide strands and also removes primer. And lastly, the DNA ligase which finally seal the nick in the lagging strand. Now, here are some differences in eukaryotic and prokaryotic DNA replication, but almost all the processes are same except few of the processes, and these are multiple replication origin in their chromosome. Eukaryotes have multiple oocytes, and the oocyte, all the oocytes will help in initiating the replication, but in case of prokaryotic cell they have a single oocyte from where the replication is being started. More types of DNA polymerases with different functions. As we learned, in case of prokaryotic cells there are only 5 types of DNA polymerases, DNA polymerase 1, 2, 3, 4 and 5, but in case of eukaryotes more than 10 polymerases had been identified yet. But out of this many polymerases, only few involved in DNA replication, and they are polymerase alpha and polymerase delta. And the most important, nucleosome assembly immediately following DNA replication. As we already learned in my earlier session, DNA in case of eukaryotes, DNA are packed with the help of nucleosome, with the help of histones, proteins. Now, what happens to these histones during replication, and what about after replication, whether the histones again combine with the DNA to get it packed or something else is there. So, during replication these nucleosomes, these histone octamers are being disassembled and then again they reassembled after the formation of DNA replication. So, origin site in eukaryotes, if we talk about yeast, they have autonomously replicating sequences, that is ARS, which enable the DNA to get it replicated, and to this ARS sequences a multi-protein complex, that is origin recognition complex, binds and unbind the DNA, and further the process will be same as that of the prokaryotic DNA. Now, in case of eukaryotes, DNA polymerase alpha which contain primase activity. Primase means it will synthesize short stretch of oligonucleotides to provide 3' end to the DNA polymerase to initiate the replication, and it also DNA polymerase also help in DNA synthesis in nucleus. And after DNA polymerase alpha has laid down from 30 to 40 nucleotides, the further replication will be completed by DNA polymerase delta, and the process is same as that of the prokaryotic DNA replication. But an important part of DNA replication in case of eukaryote is the DNA synthesis at the end of chromosome. As we know, in case of eukaryotic DNA, they have one end which will be having a gap because of the removal of the primer. Now, what about this gap? If will it be remain as gap or it will be filled up? And if it is remain in the form of gap, after each cycle of replication, the size of the DNA will be get shortened and eventually the cell will die. But it is not happening like this. Then what is happening to them? How these gaps are being filled up? So, let us discuss here. This is the end. Here we have 3 end and here we have 5 end, 3 end and 5 end. At this 5 end, an enzyme called telomerase will bind. This telomerase is also having short stretch of RNA oligonucleotides. These are short stretch of oligonucleotides. Now, these short stretch of oligonucleotides will act as a template for this 3' end, and this 3' end will be extended further. Again, this telomerase will move ahead, will provide the template, and this 3' end will be elongated. And these 3' elongated DNA strand will form a hairpin structure like this. Now, this hairpin structure when get folded, it provides 3' end, and with this 3' end, the another strand will be copied and that gap will be filled up, and this is how the telomerase will help the telomers, the end of the chromosome, to get replicated. So, let us summarize all the concept which we learned in today's session. Replication is semi-conservative, that means both the strands of the DNA will get separated and then they will be copied. Replication begins at sequence called ori site, that is origin of replication site. DNA synthesis is initiated by short segments of RNA called primers. The elongation of DNA strand is always in 5 to 3' end. New DNA synthesized from dNTPs. Replication is continuous on leading strand and discontinuous on lagging strand. New nucleotide strands are made complementary and antiparallel to the template strand. Replication takes place at a very high rate and it is very accurate because of precise nucleotide selection, proofreading and repair mechanism. Now, let us think over it. Why DNA replication is required? If suppose DNA replication is not there in the cell, inside the cell, then what will happen? Think over it. Which enzyme is responsible for polymerization of DNA? We have learned various types of enzyme which involve in different activities. So, out of those enzymes, which enzyme is mainly help in polymerization of DNA or DNA replication? What is the direction of DNA replication and why? If you know the direction, you should also know why that direction only, why not the another direction, and think over it. And the last question, what are Okazaki fragments? So, with all these questions, we will meet in our next session, and that session we will learn about transcription also along with the answer of this question. Thank you very much.